During the blocking step, we can go ahead and get our primary and secondary antibodies ready. So I'm just going to add 5 mils of our blocking buffer to each of these 2 tubes. One is the primary and one is for the secondary antibody dilution. And then we're going to add detergent to these. And this is a 20% solution of tween 20. And I'm going to do a 1 to 100 dilution into each one of these tubes of blocking buffer. That'll give us a final of 0.2% tween. So now we're ready to add the primary antibody, and we're going to do a 2-color blot today. And one of the antibodies is going to be a rabbit, and the other one is going to be a mouse. So I'm just going to add the rabbit and the mouse antibody to the same tube. And then we'll wait until the wash step, the second wash step, to add the secondary antibody. Once the blocking step is over, we're going to pour off the blocker and we're going to set it up and let it drain. I'm going to take the primary antibody and mix it really well. Pour off any excess of the blocker, and because the only difference between this and the blocker is the antibody and the detergent, I don't need to do any washing, and I'm just going to add this to the side of the membrane and let it incubate for another hour with gentle agitation. After the hour incubation, we're going to wash the membrane. We're going to pour off the primary antibody. This time I'm going to add to the side of the membrane some of the wash buffer. It's 1x PBS with 0.1% tween, and I'm just going to add a little bit of the wash buffer, and I'm going to pour off most of the primary antibody, and then I'm going to add a generous amount of the wash buffer, and then I'm going to let that agitate for 5 minutes, 4 times total.